Well, it's a real pleasure for me to introduce today's speaker, Dr. Carl Pilcher. Going sort of reverse chronological order, he was appointed director of NASA Astrobiology Institute in 2006. Prior to becoming NAI director, he was a senior scientist for astrobiology at NASA headquarters. Dr. Pilcher's scientific career was mostly conducted when he was a member of the faculty at the University of Hawaii. His significant achievements include the discoveries of water frost in Saturn's rings and on the 3 of Jupiter's Galilean satellites, and of weather on Neptune. Dr. Pilcher received his Bachelor of Science degree in chemistry from the Polytechnic Institute of Brooklyn and his PhD in chemistry and planetary science from MIT. He also holds a Master's in Public Affairs degree, Master of Public Affairs degree in International Relations from Princeton University. So he has quite a few degrees, and he's done quite a few significant things in his life. Today he's going to do a very special thing. He's going to talk about the scientific career of one of the greatest scientists of the 20th century. The title of his presentation is Explorer, Nobel Laureate, Astrobiologist, Things You Never Knew About Barry Blumberg. Would you please give a warm welcome to Dr. Carl Pilcher.
Thank you very much, Stephanie, and thank you all for coming. I see a lot of card-carrying friends of Barry in the audience, and it's great to see you all, and thanks for coming to hear what is truly a remarkable story. Even as a friend of Barry, there are many things that Barry didn't talk about a whole lot to his friends. He did not talk about his accomplishments. He was a genuinely humble man. If you asked him, you could get him talking. But if you just met Barry and were talking with him, chances are you'd wind up talking about something that you were interested in, because Barry had an insatiable curiosity. And Barry would generally ask you what you were interested in, and you'd quickly find some common area of interest. And you'd find yourself talking about movies, for example, which Barry was very interested in, which many people are, or camping or hiking, or a variety of other things. You wouldn't find out about Barry's accomplishments. And what I would like to do today is share with you the story of Barry's accomplishments, how he accomplished the things he did, which were really quite extraordinary, and relate those to what we know about Barry's NASA career, which began at the rather advanced age for Barry of 72 when he came to NASA. And what you will see is that what Barry did as the founding director of the NASA Astrobiology Institute was in fact directly traceable back to his experiences in medical research. So let's get started. Now, a lot of what I'm going to tell you, Barry wrote down in his autobiography, The Hunt for a Killer Virus Hepatitis B. And if you want to know more of the details of Barry's story, I encourage you to get the book and read it. There are other biographies of Barry available, not so much in writing, although he has an expanded biography on the Nobel website that is really very good and complements this one. It was written 4 years later, and there's a wonderful 6 or 7 hours of video, which is basically an extemporaneous biography that's available on the web, and I can tell you about that if you're interested. So, Barry's early years. He was born in New York City. He was born in New York City, the grandchild of Eastern European immigrants. He had a Jewish parochial education prior to going to high school. He was at the yeshiva of Flatbush, which is Flatbush is an area in Brooklyn. And his studies included, as is the custom in Jewish parochial education, studies of the Torah and the Talmud, which are commentaries on the Torah that have been written over many centuries. And what Barry noted in retrospect was that the Talmudic discourse, this fact-based argumentation, was very good training for his later scientific career. It started him in the mode of looking at what the facts are and then debating with others about what those facts might actually mean. And as you'll see, of course, that was a central part of his scientific career. He graduated from Far Rockaway High School, which is on a barrier island, basically, at the south end of Long Island. The graduates from that high school included, remarkably, two other Nobel laureates, Richard Feynman, who won the prize in physics in '65, and Burton Richter, who won the prize in physics the same year that Barry won the prize. And in fact, Barry said that the intellectual atmosphere in his high school was so great that he didn't encounter its equal until he arrived at Oxford for his PhD after going to med school. So it must have been quite a remarkable place at that time. He entered the Navy in 1946 at age 17. The Navy paid for him to complete a degree in physics at Union College in New York, and he was commissioned a line officer and served for 3 years, I guess it was, rising to the rank of captain of small amphibious ships. And there is Barry at age, roughly 22, sitting there enjoying life. And he said that he always appreciated the lessons he learned in the Navy: responsibility, forward planning, the importance of logistics and infrastructure. Sorry for the little font problem there. Barry looked back on his time in the Navy as really a very important time of training for him, because one of the things that he learned in the Navy was that you need to start off with plan A, but you'd better have plans B, C, and D ready in case plan A doesn't work out, because you're going to need plans B, C, and D really, really quickly in that case. And as you'll see, so much of Barry's research was done in the field, in many, many field trips. And in field trips, the unexpected was happening all the time. And as you'll see, Barry went to some pretty remote and primitive places in the world to do scientific research. So Barry always had plans B, C, and D ready, just in case plan A didn't work out. And I suspect plan A probably didn't work out an awful lot. So let me talk a bit now about his early field work and medical anthropology. So in 1947, Barry was in graduate work in physics. He had gotten out of the Navy. And he entered Columbia University Medical School. Now, it was interesting. He left graduate work in physics because his senior professor and advisor told him that he really didn't have the specific intellectual skills necessary for success in physics. And Barry said that he actually came to agree with that assessment. And apparently his father agreed with the assessment as well. So at any rate, the advice was, Barry, go to medical school. So Barry went to medical school. When he was a third-year medical student, he had the opportunity to go to Suriname, which is a country in sort of the north-central part of South America, which was then known as Dutch Guiana, and French Guiana is just to its east. Barry was, in some ways, a man of his times, and the times associated the tropics with romance. They were mysterious, and there were all these movies which we now recognize were not politically correct, but at the time were very, very popular. And Barry was really fascinated with the tropics. He was also, by this time, very deeply committed to medicine and to providing health care. And for him, this was really a fabulous opportunity to pursue both of these interests simultaneously. So he spent the summer in Suriname. His job there, his role, was to provide basic medical services, kinds of things that you could imagine remote populations needing, but also to study disease. And he was based at a bauxite mine, an aluminum ore mine in the city of Moengo, which is not far from the coast, but you have to go up a fairly major river to get there. This was a mine that had produced most of the bauxite ore that supported the Canadian effort during World War II and a lot of the U.S. effort during World War II. So there was, you could get large ships into the mine, and you could export the ore out. Now, Suriname was very interesting from the standpoint of populations because there were, of course, natives of South America in Suriname. Suriname, like many countries of the New World, had a slave trade for some years, but it was also the site of one of the first successful slave rebellions in the mid-18th century. So there were also a number of descendants of those slaves, who formed a very large population of African origin. There were also Europeans, particularly from Holland and Northern Europe. Whoops. Indonesians, Indians, Chinese, etc. And what Barry found in studying the prevalence of disease, and malaria and filaria, two parasitic diseases, malaria, of course, a protist, and filaria, the parasite is a worm, particularly nasty disease. What he found was that the susceptibility to infection varied greatly among those different populations. Now, these were people who were living in the same place at the same time under the same conditions. And yet some people got sick and some people didn't get sick. And Barry wondered why. Now, just to illustrate sort of what Barry was prepared to do at that time, this is a trip that they made on the Marilyn River during that summer. This was to get into the far more remote interior of Suriname to do a study of the prevalence of filaria and malaria in really remote areas of Suriname. So Barry was really prepared to do things at the time that were really beyond what most people were doing, certainly most researchers, scientific researchers. So this was really a seminal period for Barry. It was during this period that the central question that motivated his entire scientific career was really formed. And that question is, how do inheritance, human behavior, and the environment interrelate in the context of disease? And he wrote in his book, "It was in Suriname that I learned to rely on observations in the field. New observations led to new hypotheses that could not have been induced by laboratory-based experiments. The field work was in turn very dependent on laboratory techniques, and hypotheses were confirmed or rejected by experimental testing." This, too, was a way of approaching science that characterized Barry's scientific career. The use of field work, theoretical work, that is the development of hypotheses, and then the testing of those hypotheses in the laboratory, and the interrelationship between those three modes of investigation were also central to Barry's work. So after med school, Barry trained at Bellevue Hospital, and anybody who is familiar with New York City, and particularly New York City at that time, knows that Bellevue Hospital was one amazing place to train in medicine at that time, followed by going up to Columbia Presbyterian Medical Center uptown. And again, what he found, particularly in the population that was treated at Bellevue, which was a city hospital, and this computer is very touchy here, he found again that some populations, some demographic groups, Scandinavians for example, were much more likely to contract tuberculosis, which was very common in New York City at the time, than were other populations. Again, reinforcing the observations that he had made in Suriname. From 1955 to 57, he was at Oxford doing a PhD in biochemistry. And during that time, he developed a very specific interest in genetic polymorphisms, basically variations, genetic variations, and their relationship to disease susceptibility. Now, the thing that is important about a genetic variation that is related to disease susceptibility is, of course, you can determine that before somebody gets sick. So that means that if you know who is susceptible to a particular disease, there is the potential, at least, for intervention. For example, if you know that you are genetically susceptible to heart disease, you may spend more time really focusing on keeping your cholesterol down. Very simple, not so simple sometimes, intervention. But that's the kind of thing that motivated Barry. So he wrote that it began to dawn on me that the study of human polymorphisms would provide a conceptual framework for the investigation of inherited human variation and its connections with disease and survival. Now, that concept of determining a susceptibility to disease before someone gets sick is basically preventive medicine. Well, preventive medicine was not a hotly pursued topic in the medical community at the time, and some would argue even today. It certainly wasn't a high-status professional goal. I mean, after all, what happens when you are successful in preventive medicine? nobody gets sick. Try imagining a TV medical drama based on preventive medicine. I don't think it would do too well in the ratings against ER or, in the Oscar competition against Contagion. You're not going to find too many people getting excited about the results of preventive medicine. At the same time, the particular thing that he was studying, human polymorphisms, at the time were also seen as sort of ho-hum by the medical community. They were used in forensics if people committed a crime, as we see on CSI now all the time. If there was a paternity suit, blood banking was concerned about human polymorphisms, make sure that blood types were the right type. Anthropologists were concerned about human polymorphisms because it enabled them to trace populations. So Barry was pursuing a not very medically exciting, or at least as seen exciting by his contemporaries, not very exciting medical field, preventive medicine, using a sort of ho-hum, again, as seen by the medical community, technique, studying human polymorphisms. Worse, to study human polymorphisms, you have to do population studies. Well, population studies, of course, are much less precise, as viewed by the traditional medical community, than reductionist medicine, which looks at the chemistry and the physics of what's going on inside the body. But Barry pressed on. This was Barry's nature. He pressed on in face of this skepticism and did that work for several years at the National Institutes of Health before he moved to what was then called the Institute for Cancer Research, which is now known as the Fox Chase Cancer Center. And he talked about that move from NIH. And he said his main problem at NIH was fitting what I wanted to do into the discipline-determined rigidity of the constituent institutes. Then and now, NIH comprises about 20 individual institutes, each devoted to a particular disease or category of disease. And of course, having that structure makes it very difficult to work across the boundaries of those institutes. Barry's research, as he says, ranged over several disciplines: population anthropology, epidemiology, environment-host interactions. There was, of course, a very major clinical component to his work. And even though the Institute for Cancer Research was dedicated to cancer, we were never asked what relation our research had to immediate cancer applications. So he found a freedom to work across disciplinary boundaries at ICR, now Fox Chase. And when one thinks about astrobiology, which, as most of this audience knows, is extremely interdisciplinary, it's not surprising to find that later in life, when Barry had decided not to continue as an active medical researcher, that it was astrobiology as another science that really got his attention. So, how did Barry come to the Nobel Prize? One thing I can tell you for sure is he must have been as surprised as anybody that this is where his path took him. So, the question he was focused on: what determines who gets sick? In 1955, there was a new technique developed, gel electrophoresis. Now, anybody who has taken elementary biology, even in high school, thinks of gel electrophoresis as, oh my, everybody does that. You do it in high school, you do it in freshman biology. But however, somebody had to develop it. There was the technique before that was paper electrophoresis. So this was a newly developed technique in 1955, and Barry and his colleagues began an extensive study of genetic polymorphisms, about the distribution of different variants of genes in populations living under very different environmental conditions. Again, getting at what's the interaction between behavior, the environment, and your genetic makeup with regard to susceptibility to disease. This was a time that his field work really took off, and he was traveling literally to the ends of the earth. He started close to home. He actually started around Oxford, getting a baseline population, and went to the Basque Country, because it turns out the Basque people actually have a fairly distinct population. Then to Nigeria, and over the next several years to northern Alaska to study the Inuits, Greece, Pacific Islands, Native Americans, South America, etc. What they generally did was they tagged along with other studies which were collecting blood samples, and so they would be able to get a portion of those samples that were collected without causing any additional interventions in those populations. And they had their general conceptual framework that by understanding the distribution of genetic polymorphisms, they would learn something, and doing this in different environments. They would learn something about disease, but they didn't know what they were going to learn. It was way too early to postulate that we're going to study this disease by studying these genetic polymorphisms. So what they were really doing was exploring. In this case, the space that they were exploring was human genetic variation. And they were exploring for what they were going to find about the associations between environment, those human genetic variations, and disease. So I would argue that Barry was a true explorer from the outset. This is a picture of Barry taken in Suriname during this time. Here he is in northern Alaska. This is during the Inuit study, one of the Inuit studies, and he is dressed in this case for a meeting with village leaders. And here he is in the marvelous picture that we used on the poster, which was taken in Norway in the summer of 1963. So they had quite a bit of success with this, and they discovered they found, got more information about a number of previously known genetic polymorphisms. But what they wanted was they really wanted to make a splash. They wanted to find a new polymorphic system. Now, to give you an idea of what they were looking for, one of the most common known polymorphic systems is the blood type system, whether you're type A, type B, type O, Rh negative, Rh positive, and it turns out that there's a whole bunch of other variants of blood types. And this forms a polymorphic system. You can trace those variants back to genetic variations. And so what they were looking for was they were looking for a similar system, but one that was associated with disease susceptibility. And they developed a new technique to do this. And I really wish that Barry were still here so I could ask him, how did they think of this? This was really a very clever thing. They decided to study the blood of patients who had received numerous blood transfusions. Now, the idea here is that when a patient receives a blood transfusion, that patient may receive a blood protein that, of course, the patient has because we all have basically the same proteins, but might receive a slightly different variant of that protein as the result of a genetic polymorphism. That is, the person who donated the blood has a different version, a different allele of the gene responsible for that protein. Well, if that protein was different enough, it might be an antigen. That is, it might be a protein that stimulates an immune response in the recipient. So the immune response would involve the generation of an antibody. Now, so we're going to be talking a lot about antigens and antibodies, and the simplest way to keep them straight is to remember that antigen derives from antibody generator. So the foreign protein introduced into the transfused patient serves as the antigen, and if it is antigenic, then the immune response may produce an antibody. So what they did was they did a double diffusion using gel electrophoresis, using agar gels, and what they did was they would put the serum, which is the liquid component of the blood with all the white cells and red cells and clotting factors separated up. They'd put the serum from the transfused patient in a central well, and they would put the sera from the test subjects in adjacent wells. What they would do is that they would then run the gel, and if the test subject sera contained a protein, that is an antigen, for which the transfused patients, for which the transfused patient's serum contained the antibody, the antigen and the antibody would bind. That's how antibodies deal with antigens, and they would precipitate. And you could actually see that precipitate. It would form an arc, and you could either stain it, or sometimes you could just see it directly. And that arc could then be visualized and studied. Well, it turned out that they did discover a new polymorphic system. They called it the AG system, And it turned out to be a system in low-density lipoproteins that were correlated with susceptibility to heart attack, stroke, and diabetes. So that was one of the things they pursued. But they, just after they discovered that system, they discovered something that really piqued their interest. And I'm just going to jump right to that because that follows the line of our story. They discovered something that they called the Australia antigen. Now, they called it the Australia antigen, one, because that was a nice neutral name, and two, because it was first observed in the blood of an Australian aborigine. They discovered other antigens at the time that they called the Pennsylvania antigen and a variety of other similarly neutral names, and they didn't pursue those. And Barry wondered in his book what might have happened if they had pursued those. Why did they pursue the Australia antigen and they didn't pursue the others? You know, would the world have turned out differently? And perhaps, but we will never know. So, the Australia antigen. As I said, it was discovered first in the serum of an Australian aborigine, but it was then found to be common in populations in Asia, the Pacific, Africa, Eastern and Southern Europe, but not in the US and not in Western Europe. That is, it wasn't common. It wasn't non-existent in those other populations, but it wasn't common. Now, again, remember, they're looking for genetic polymorphisms. They're not looking for infectious disease at this point. And so the first thing they do is they analyze their data with an inheritance model. After all, they had just found a polymorphic system. And the data seemed to be consistent with an inheritance model, but there were some surprising exceptions. The kinds of exceptions would be cases, they started off doing family studies. And so in family studies, what you expect in an inheritance model is if both parents have the putative gene for the Australia antigen, then you expect all the children to have it. And in general, that was the case. If one parent has it, then you expect it to follow a dominant gene, recessive gene model, and you would, the children would have it depending upon how they inherited the genes from the parents. And they could interpret the data in terms of this model, but it didn't always work. So one of the things that they noticed is that most of the sera that contained Australia antigen were also from patients who had been transfused. And so now they found themselves with this curious observation that they were looking at patients that had been transfused in comparison to patients who had been transfused. Now, the suggestion here was that, well, if AU is associated with transfusion, maybe it's a blood-borne infectious disease rather than inherently a genetic disease. And they started getting other data that led them down that same track. They were also doing normal population studies. That is, studies, again, they were piggybacking on other people's work in many cases. Just studying background populations. They were doing one in the South Pacific. They were doing one in Georgia here in the US. And they found rare instances in some of these normal populations of AU-positive individuals who had the Australia antigen. And in one case, it turned out that that person, which was one person in a study of several hundred, had just been diagnosed with a case of hepatitis. And in another case, somebody who had not been AU positive became AU positive, and it turned out that they had just received a transfusion. So again, they were getting clues as to what AU, the Australia antigen, might be. They also found that there was a correlation with patients at high risk for leukemia. For example, people who survived the atomic bombs in Hiroshima and Nagasaki, people who were exposed to radiation during atomic testing in the Pacific, and Down syndrome patients. Now, the study of Down syndrome patients proved to be very, very informative. A bit of history. Prior to 1950, 1960, the medical advice to patients, parents who had a Down's child, was to institutionalize the child. Now, obviously, practice today is very different, but this was the practice in the first part of this century. And so there were, by 1950, huge institutions with thousands, in some cases, of Down syndrome patients, all institutionalized. But that pattern was starting to break down in the 1950s, and by the mid-1960s, a lot of these institutions had been at least partially dismantled, and there were a lot of Down syndrome patients in group homes, in smaller group environments. So what they had was they had a population which had something in common, a genetic-based disease, Down syndrome, but they had them in environments where there were 1,000 or more people interacting on a daily basis, and environments where smaller and smaller groups were interacting. And what they found was the frequency of the Australia antigen, which was fairly common among Down syndrome patients, was inversely related to the institution size. In other words, they were more likely to find AU-positive individuals in the large institutions than they were in the small group homes. And again, this is consistent with an infectious model. That is, that the more people you have to pass the infectious agent around between, the more likely you are to find individuals with that infectious agent. And they continued to find other individuals, just surprising results, which I don't have the time to go into. So the surprises continued to associate Australia antigen with hepatitis. So this led to them deciding, let's not do more family studies and more studies of populations related genetically. Let's start looking at people who have clinical cases of hepatitis and understand what relationship the Australia antigen has to those populations. And by mid-1967, they had documented the association of the Australia antigen with hepatitis. So what do? They write a paper reporting this result, and the paper gets rejected by the journal Annals of Internal Medicine. Why did the paper get rejected? Well, there were several reasons. First of all, no one on the team was a virologist. Hepatitis was a disease that had been known for many, many years, yellow jaundice. The symptoms of hepatitis are very, very obvious. There was a well-developed community that had been studying hepatitis for decades. They had been searching for the infectious agent of hepatitis for decades, and they hadn't found it. Now, here came along people who were basically clinical physicians and some biochemists and a few laboratory spectroscopists. They had no formal training in epidemiology. None of them was a virologist, and none of them were a member of the hepatitis research community. So that was part of the problem. This was about the time that Barry read Thomas Kuhn's The Structure of Scientific Revolutions, and he began to get some perspective. Those of you familiar with the book, and I suspect that's many people in this room, know that what Kuhn argued is that a scientific paradigm develops in a community, and it's a model, and the community basically endorses that model, and that becomes the paradigm of that community. And when data first start to appear that tend to undercut that model, contradict that model, the first reaction of that scientific community is to resist this change. They're not going to change their model, their paradigm, because a little data here is inconsistent. There's always ways to explain why a little data are inconsistent. And what has to happen is the evidence that the model is not a correct model has to build up to a point, and there's resistance up to that point. And when the contradictory data reach a critical point, then a change occurs very rapidly, and that change Kuhn referred to as a scientific revolution. And what Barry began to conclude was that they were on the cutting edge of a scientific revolution. And of course, he was exactly right. The reviewer, the principal reviewer of the paper, later told Barry that the reason he had rejected it was that there had been so many erroneous claims of discovery of the serum hepatitis, which is what hepatitis B was called at the time, the serum hepatitis virus, that the reviewer just didn't want to see another erroneous claim in the literature, which was only, bound to be retracted someplace downstream. Barry's reaction to this was very interesting. If such rejections are taken too seriously, he wrote, they can lead to an attitude of martyrdom and of opposition to a recalcitrant establishment. We, of course, know of no colleagues who ever responded that way to the rejection of the paper. Our unhappy sentiments didn't last, and the rebuff didn't slow us down. Again, this is Barry. Press on. Don't worry about that. Just keep doing what you're doing as long as you're doing the right thing. Now, they had written an earlier paper before the main paper, submitted to the same journal. The earlier paper didn't make as strong a claim, and the earlier paper actually got published. But it still remained to be demonstrated that the Australia antigen could be equated with the hepatitis virus. So at this point, they knew that there was an association, but they didn't actually know what the Australia antigen was. Now, at this point, they did something very important. Let me skip down to the quote from Barry's book. One of the most important steps we took was to distribute vials of the anti-serum, that is, the antibodies, and the antigen to investigators who requested them. With these, it was very easy for scientists, even in laboratories with rudimentary equipment, to repeat and extend our studies. Within a few months, there was confirmation from other investigators, often from parts of the world where hepatitis B was more common than in the United States. So, in essence, what Barry did was to practice science in the most open and generous way. Give his reagents that he and his team had spent a lot of time, a lot of years of their lives developing. Just give these reagents out freely to the scientific community, to anybody who asked. So what resulted was research started going on all over the world in many, many different laboratories. And it was that research with Barry's team contributing. But at this time, by this point, Barry's team really wasn't really that much in the lead on this final stage in the research. They were in the lead on direct imaging by electron microscopy because they had one of the world's leaders in electron microscopy, which again was a relatively new field, at Fox Chase Cancer Center. And they finally demonstrated that the Australia antigen is not the hepatitis virus, but it is a component of the virus. So here is the hepatitis virus. It is a very simple virus in its structure. It is far from a simple virus in its action. It has four main components. The surface antigen, and this is hepatitis B, S is for surface antigen, is the protein coat on the outside of the virus. That turned out to be the Australia antigen. And from here on out, we'll talk about HBsAg rather than the Australia antigen. That covers a core protein, which is called HBC for core, Ag for antigen. And within the capsid formed by the core protein, there is DNA polymerase, an enzyme that will form the DNA polymer, a reverse transcriptase, which is an enzyme that will make DNA from RNA, because it turns out that although the hepatitis B virus contains DNA, there is a stage in its life cycle in which it reverse transcribes RNA to DNA. So it's actually a retrovirus like HIV, even though it contains DNA. And the actual DNA of the virus itself, which contains 4 genes, just 4 genes. Very simple. However, the way it behaves and the disease that it causes is far from simple. This is a micrograph of HBV particles. And most of the particles you see are not the virus. All of these particles, the circular particles, and these are about 21 nanometers across, and the elongated particles that are the same diameter but of varying length, those are all the surface antigen. Those are all the protein that coats the virus. There are actually only 3 whole viruses in this image. One is here, one is there, and one is there. And you can see that they contain a central portion, and that central portion is filled, in this case, with DNA. Now, one of the things you see immediately in this micrograph is there is much, much, much more of the surface antigen than there are of the live or the whole virus. And that is a characteristic of hepatitis infection. The abundance of surface antigen relative to the abundance of live virus is typically 1,000 to 1. And that turns out to be key for one of the things that Barry did next, which was the development of a vaccine, Barry and his team. So we'll get to that part of the story in just a second. But the first part of the story, the next part of the story, is the issue of the transmission of hepatitis through blood transfusions. As you can see from the studies in which they found themselves studying transfused patients against transfused patients, hepatitis is readily communicated by blood transfusions. Before this work, there was no way of detecting hepatitis in the blood system. So there was no way to test donated blood for hepatitis virus. At the same time, there were more than 150,000 cases a year of post-transfusion hepatitis, that is, hepatitis caused specifically by blood transfusion. So the first application of their discovery was screening donated blood. All they had to do was screen now for the surface antigen, which was present in such high abundance that it was actually very readily detectable, even though they weren't using very sensitive techniques. There were much more sensitive techniques that they could apply. They were just more expensive, more difficult to do in the field. And so they kept doing the double diffusion gels for a long time because it worked extremely well. But it took 3 to 4 years before testing blood, donated blood, for the surface antigen became adopted. Why? Again, residual skepticism. You know, these guys say that they can stop the post-transfusion hepatitis, but you know, people have been trying to do that for a long time. Why should we believe them? There was also an economic motive. After World War II, with the development of better anesthetics and better antibiotics, there was a huge growth in the number of surgeries, open heart surgery, kidney transplants, etc., cancer surgery. And there was a great demand for blood. And so there was both a nonprofit blood bank industry and there was also a for-profit blood bank industry. And of course, they all have their economic models. And if somebody says you're going to have to start throwing away an awful lot of your blood, plus you're going to have to do all these tests, suddenly that affects the economics. And so it really took the University of Pennsylvania Hospital, which was the first to institute this, because Barry, of course, was associated with the University of Pennsylvania. It really took them instituting it, demonstrating that they greatly reduced the incidence of post-transfusion hepatitis before other hospitals had to adopt it. Because if they didn't adopt it, they would get into the situation of they could be sued for malpractice if there was a well-demonstrated mechanism of preventing it. So that took about 3 to 4 years, and that finally happened. And it greatly reduced the transmission of HBV in blood transfusions. And then that process continued when HCV was discovered in the 1980s. So, but the next step was to develop a vaccine to prevent the infection by other routes. HBV is extremely infectious. It is readily transmitted. It's more infectious than HIV. It's transmitted mother to child at birth. It's transmitted sexually through contaminated needles, sharing common personal items. There are even cases of it being transmitted by sharing a toothbrush. Blood-to-blood transmission is the transmission mode. If somebody has bleeding gums, they use a toothbrush. Somebody else uses that toothbrush. They also have bleeding gums. You can have blood-to-blood transmission. There was a case in a hospital early on in the days of development of computers in which they installed an IBM punch card system. And it was transmitted through paper cuts from the IBM punch cards. So it is a very, very infectious agent. It infects about 1/4 of the world's population even today, about 2 billion people. It's endemic in China. And it also causes most primary cancers of the liver, which kills about 1.5 million people annually. And in doing their research, what Barry and his team found was that the surface antigen was the protective agent. That is, that if you inoculated somebody with a very small amount of the surface antigen, that person would develop antibodies to the surface antigen. If that person was later exposed to the live virus, those antibodies would attack the virus and provide protection. And here's where that 1,000-to-1 ratio of surface antigen to live virus comes in, because they could collect 300 milliliters, for example, of serum from an infected patient, and that could be used to, after processing, to inoculate many, many individuals. You didn't need a lot of the surface antigen to generate the antibody response. So they developed a vaccine based on that, actually using the blood products from infected individuals, separating out the live virus from the surface antigen, purifying the surface antigen, adding some boosting agents, and then using that as the vaccine. It is the first and only vaccine that has ever been developed by that method. And fundamentally, it's the same vaccine that's being used today, although it's no longer made from blood products. It's made by recombinant techniques, cloning the gene for the surface antigen into E. coli, basically. So the blood-derived product was effective and safe, but there was still some reluctance to use it because, in principle, if you're using a blood product, there could be live virus. The recombinant technique that was developed in the early 1980s fixed that, and the vaccine is now one of the most widely used vaccines in the world. Over 1 billion doses have been administered since the modern vaccine became available. 177 countries, as of a couple of years ago, it may be higher now, have national vaccination programs. It's the first vaccine that prevents a cancer. There's really a major human cancer. There's really only a second now, the vaccine against cervical cancer, which prevents infection by the human papillomavirus. It's second only to smoking prevention, smoking prevention as a cancer prevention program. It saved millions of lives and had many other highly beneficial effects. There's a story I'd love to tell you, but I notice I'm running out of time, so I'm going to move on. This is Barry at Oxford University in 1973. The chart behind him actually traces out all the details of this story. I've obviously just summarized some of the highlights in the story, but the story was a very, very intricate story involving field work and populations all over the world. And that chart is kind of the outline of Barry's book. Here he is receiving the Nobel Prize, 1976, with the King of Sweden. It was a remarkable collection of individuals that year. Burton Richter, who got the prize in physics, Barry's high school mate, although at a different time. Saul Bellow got the prize in literature. Sam Ting of the instrument that was just brought up to the International Space Station. Milton Friedman got the prize in economics. William Lipscomb, a very famous chemist, got the prize in chemistry. And Carlton Gajicek, who was the co-winner of the prize in physiology or medicine along with Barry. Gajicek studied Kuru disease in the Fijian Islands. There were a lot of happy people at the Fox Chase Cancer Center. And that is Barry's wife, Jean. China. Hepatitis is an extremely serious problem in China. It was at the time, and it remains a very serious problem in China today. In the mid-1970s, China's population was about 1 billion, and they had the highest incidence in the world of HBV infection and the highest incidence of primary cancer of the liver. The proportions of populations in different parts of China that were infected with hepatitis B at the time was in the mid-teens, typically. That is, the mid-teens, 15, 16% of the entire population. In some areas of China, that remains the case today. The prize, as well the same year of the fall of the Gang of Four, led to an invitation for Barry to visit China. It probably wouldn't have happened before either of those two events. At this time, Western visitors were still rare, and foreign technologies were viewed as politically suspect. He basically was led on a tour of China, went to many of the major cities, spoke to audiences in the thousands, and basically told them about the development of the vaccine, what they could do to address this tremendously serious problem in their country. He helped them acquire the rights to manufacture the vaccine and to begin a nationwide vaccination program. In 1978, he did a similar visit to Taiwan, which had a similar impact. And he later referred to the 1977 trip as the most important field trip of my life, a life that has included many field trips. He has since been very widely honored for his contributions to healthcare in China and to Asia more generally. Here, he and Jean are in China in April and May of 2002. This is a tree planting ceremony, part of a very long trip on which he was lavished with praise for the positive impact he had in China. And here he is in 2009, right here in San Francisco, at the Chinese hospital at an event. He was being awarded the 3rd annual Hep B free, Hepatitis B free Superhero Award, and the award included a cape with that emblem on it by the San Francisco Hep B Free organization and the Chinese Hospital of San Francisco. Very quickly, because I can see that I'm running out of time, somewhat later in his career, he went back to Oxford, to Balliol College, the college where he had gotten his PhD. He had been there earlier. He served as Eastman Professor in 1983. Balliol is one of Oxford's oldest colleges. It's known particularly for political science and philosophy. It's not terribly well known for science. He was the first American to be elected master. Also the first scientist, if you don't count a 14th century alchemist. And he served for 5 years. He said that the master of the college had no power but a great deal of influence. And then an interesting observation. Learning how to lead without actual power came in good use in later years when, for example, at the NASA Astrobiology Institute, I had considerable administrative power, but only rarely had need to use it. This is Barry in his Balliol robes, taken just in February of this year, just a couple of months before he passed away, along with some friends that you can see above him. This was taken in Ben Franklin Hall in Philadelphia. Ben Franklin Hall is the home of the American Philosophical Society, which Barry was president of from 2005, I believe, until he passed away earlier this year. So we come to Barry and astrobiology. I put in a couple of slides about astrobiology just in case there was anybody in the audience who didn't know what it was. I don't think there are many such people in this audience. So, you know, astrobiology's 3 questions. It involves 4 areas of science: the understanding of the nature of life on Earth, its diversity, its ubiquity. We find it basically everywhere we can. Computer's awfully touchy. And its antiquity. We find it in, evidence of it in 3.5 billion-year-old stromatolites. Another area is the environments that we are discovering on other planets in this planetary system, in our solar system, and on planets around other stars, and the prevalence of the raw ingredients for life in interstellar space and in meteorites being delivered to these habitable planets. So these are 4 very diverse areas. It requires the expertise of people in the biological sciences, the geological sciences, and the astronomical sciences. A highly, highly interdisciplinary field. And you can see from Barry's background why such a highly interdisciplinary field with such amazing questions would be attractive. So NASA comes a-calling. In 1997, Barry was invited to join Stanford's program on human biology, where he taught medical anthropology and the scientific process. Turns out that somebody in the audience, Mal Cohn, was teaching at Stanford at the time, knew Barry from Philadelphia, and Mal basically introduced Barry to NASA and introduced NASA to Barry. So Barry was invited to attend the Astrobiology Roadmap Workshop at Ames in July 1998, and astrobiology intrigued him. What a surprise. And he noted that NASA was applying scientific principles and process to questions that had been of interest to philosophers, the religious, ethicists, etc., for countless generations. Later in 1998, he co-chaired another Ames Roadmap Workshop on genomic studies on the International Space Station, and in 1999, at Dan Goldin's invitation, he became the founding director of the NASA Astrobiology Institute. There he is with Dan, and Barry, of course, served for 3 years in that role. This is also from Barry. I understood that my mandate was to establish a basic science organization that could discover and understand natural phenomena that related to early life and to life elsewhere. At an introductory address to members of the Institute's Executive Council, I told them that I did not expect them to do exactly what they said they would do in their applications, since in a fast-moving field, observations made after the application had been written could greatly change the path of research. This was greeted with cheers. Again, you can see exactly where that came from. Look at his own career. He started off looking for genetic polymorphisms and discovered the source of one of the most serious diseases that inflicts mankind. Not what was expected. This is Barry with the NAI Executive Council some years later in 2009 when he was a distinguished scientist with both the Astrobiology Institute and the NASA Lunar Science Institute. And we were having an Executive Council meeting in Yellowstone National Park. Barry continued his focus on getting out into the field. This is Barry up in the high Arctic on Devon Island at the NASA Houghton Mars project in 2000. This is Barry at the Iron Mountain Mine near Redding, California in September 2002. Barry was claustrophobic, and he went down into the mine. And he first had to overcome his claustrophobia in order to actually do it. But he was determined to see what things were all about in the field. And he went down. One of the things that developed after Barry left the NAI was a partnership with the American Philosophical Society after he became the partner. The American Philosophical Society had a program called the Lewis and Clark Program for field research, which supported young investigators going all over the world. And we formed, in partnership with Barry, a specific version of this program for astrobiology, which has sent astrobiology graduate students and postdocs, early career researchers in general, to many, many different places in the world. These are just a few, and I'm not going to go into the details here. But this is one of the many things that emerged from Barry's tenure as director and the things that followed afterward. Barry was also very concerned with the study of human behavior. Human behavior was an essential element of susceptibility to disease. It was that third component in addition to the environment and to genetic predisposition. And Barry brought an anthropologist into the NAI, Lisa Faithorn. Here she is at the NAI General Meeting in 2001 because Barry wanted a card-carrying anthropologist to study these teams that were now supposed to be competing for membership in this institute, but then they were supposed to become cooperative and collaborative afterward, and then 5 years after that, when their agreements expired, they became competitors again. And Barry wanted to understand how that was all going to work. And he figured who better to observe how that's going to work than an anthropologist. So we had an anthropologist, and Lisa remains a person closely associated with the NAI, although no longer employed by it today. Barry was ahead of his time with Facebook. He wanted to have a very early version of something that we might now recognize as a predecessor of Facebook. So we had an astrobiology photo directory 10 years ago, well before Facebook was on the scene. He introduced the concept of focus groups, bringing communities together in focus groups, and he established a virus focus group to bring virology into astrobiology where it had not been a prominent part. He clearly had a great sense of the importance of international collaboration. So the International Partners Program was also something that Barry gave a great deal of emphasis to. This is a picture taken at the signing of the partnership agreement between the NAI and the Astrobiology Society of Britain in London. This was taken at the U.S. Embassy in London in 2002, I believe, or 2001. Here's Barry with David Wynne Williams, a member of the British Antarctic Survey, who was tragically killed in a bicycling accident a few years after this picture was taken. Barry also appreciated the contributions that all people from all walks of life have to make to science. He started the Minority Institution Research Support Program, and here is a picture taken at the Astrobiology Science Conference in 2006. There's Barry with a few of us and with a number of the faculty who received support through the Minority Institution Research Support Program. Barry was also a great fan of involving ordinary citizens in science. Citizen science projects were a passion of his. In fact, this is what he was speaking about at the meeting at which he passed away here in April. As Barry said, people keep saying that basic research is just a fishing expedition. I never understood why people objected to that. After all, that's the way you catch fish. Barry caught a lot of fish in his career. We now have the Blumberg Center for Science Innovation, which is the home of the NASA Lunar Science Institute, just at the head of the parade grounds. Barry with his wife, Jean. Barry passed away within a day of his 57th anniversary, which was very poignant for everyone. Barry was just such a genuinely warm human being. Genuine is the right word to describe Barry. There wasn't a false bone in his body. He was warm, he was humble, he was generous, he was kind. A lot of people in this room knew him, and you know whereof I speak. Those who know his family know that his family, his kids, their kids, are really, really nice people. And in this case, this is just one really, really nice family. So we all have wonderful, wonderful memories of Barry. Thank you.
If you have a question, you have to raise your hand.
Carl, you quoted extensively from a book that Barry wrote, which I was unaware of. Could you give us the title and where one could get a copy?
I sure can. It is published by Princeton University Press. I presume, I didn't check to verify that it's still in print. I bought it in the Princeton University bookstore some years ago. But there it is. It's The Hunt for a Killer Virus, Hepatitis B, by Baruch S. Blumberg, Princeton University Press, 2002. Lynn. Let's get a microphone to you.
Said to me, it's typical Barry that more members of my family have climbed Mount Katahdin than read my autobiography.
I tell you, I really wish I had read his autobiography as carefully before he passed away as I have since, because I now have so many questions I want to ask him.
Thank you. Carl, you mentioned that there's an archive of video, I assume interviews with Barry.
Yes.
Where is that?
Google Web of Stories. And then when you get to their homepage, just search in their search engine on Blumberg, and you'll come to it. It's 6 or 7 hours broken up into 80 individual segments, each a few minutes long. And basically, it's an extemporaneous version of his book. He takes you from his early childhood right through his career at NAI, talks about all the things that I've talked about.
Google Web of Stories?
Web of Stories. Just
great.
Type
Thanks.
Web of Stories into the search engine, and you'll and then search on Blumberg in their search window, and you'll be there in 30 seconds.
Any further questions? All right.
Carl, thank you so much for doing this. It's just amazing to hear this backstory. And I think more of us aren't able to speak right now because we're so choked up with our own memories of Barry. But I'm going to try to push past that because I want to say something that I think is really important to this crowd. And in fact, for everybody in this crowd to take back to everyone you know. And that is that because of Barry's passing, several of us wanted to learn more about what to do when a loved one has a heart attack. And so we purchased at the Lunar Science Institute one of those AED devices. And in order to learn how to use that, we all took a CPR course just last Friday. And what we were surprised to learn is that there have been some changes that the Red Cross now advises in the way you give CPR and when you give it. And without going into what those details are, I would just like to urge every single person here to get retrained right now. Even if you think you know what to do, I can promise you some things have changed just in the last year. And for those of us that were so close to him, we all wonder and wish we knew what we know now when he had passed. So I really just can't emphasize enough that you need to get CPR trained again.
Okay, well, this is a great colloquium, Carl. I think we should give our speaker one more round of applause.
Thank you all for coming and sharing in Barry's story.
And I think we have some refreshments across the way, so please stay for that.